Hello, students. Hopefully you're starting to understand the details of this extraordinarily complex process of protein synthesis. So there's just a few little loose ends then that we need to tidy up in this last screencast. And that is what happens, the first one is what happens to those protein products once they're made. So as you probably already are aware, the first thing that happens is folding. So as the protein is being built, the interactions between its amino acids are going to cause it to be folded up right there and then on the spot. So literally as it's coming out of the ribosome, it's going to be folding and interacting in this very unique way. That's kind of crazy to think about. And sometimes it needs some help to make sure that it's folded into the right way. And so there's proteins that can do that. One example of these are what are called chaperone proteins. And these simply aid in folding. So one example is they may be able to create a hydrophobic environment for folding to be able to occur in. And so here you can kind of see a model of what this looks like. And then here's kind of a schematic. But basically, as the protein folds, it kind of gets put inside this space, and then it folds in that proper environment, and then it pops out. This always just kind of blows my mind. I don't really understand, like, really that happens in the cell? But apparently that happens in the cell. Mind-boggling. The next thing that has to happen to that protein is it has to go to the right spot. And so we call that protein signaling. Like, how do we make sure that the protein actually gets to where it needs to get? And so the key to this is what's called a signal sequence. And that is going to direct the protein. That's my signal. I'm supposed to open up the Google Meet for the tutoring center. Anyway, so there's this signal sequence that is going to direct the protein to a specific organelle. And so you can see on the ribosome here in green, there's this little tiny region, kind of the first handful of amino acids that are generated. They're actually not important structurally for the protein. Rather, they're important for making sure the protein gets where it needs to go. And so there's one particular sequence that will interact with a particular protein called SRP. That's the signal recognition particle. And the signal recognition particle is going to bind and kind of attach to that. And then it also has a binding site that will recognize and attach to a protein on the rough endoplasmic reticulum. So this particular signal sequence, if it is there, is going to cause this protein to be snatched up by this little yellow signal recognition particle and delivered to the endoplasmic reticulum. And at this point, the ribosome is now attached to the endoplasmic reticulum, and the protein is going to be produced in the endoplasmic reticulum. In other words, this is what makes the rough ER rough. All the ribosomes start in the cytoplasm doing their thing, but if this particular signal sequence is generated, then that delivers the ribosome to the endoplasmic reticulum, so the protein can be put there. And then as we know, from the endoplasmic reticulum, it can undergo exocytosis into a vesicle, be transported to the Golgi apparatus for modification, and then from there to the lysosomes or wherever else in the cell it might be. This is all important to make sure the protein gets where it needs to go. If that signal recognition particle was absent, the ribosome would just stay in the cytoplasm and it would be used there. So a glycolysis enzyme, for example, wouldn't have that signal recognition particle. It would just be produced in the cytoplasm, and then the protein would stay in the cytoplasm. Okay, the last piece I wanted to talk about is mutation, and more mutation, because we've already talked about some mutations. We last talked about DNA. So, we talked about kind of major chromosomes. Well, so we've talked about causes of mutations, right? So one is errors in meiosis that can occur, where, it could be non-disjunction, where you get a whole extra chromosome. It could be like a little missing piece of a chromosome, something like that. We've talked about how an error in DNA replication can cause a mutation. And I don't think we've talked much about it, but mutagens can also do it. So exposure to UV light, certain chemicals like those in cigarettes, X-rays, these kinds of things can cause damage to the DNA as well. So in this unit, we are now ready to understand another category of mutation, which are called base pair substitutions. This is where one or a few base pair or pairs is substituted for another. And the consequences of these are pretty obvious once you kind of see what happens. So the first type of base pair substitution is a silent mutation. In a silent mutation, the same amino acid is inserted despite a change in the DNA. So it's called silent because it has no effect on phenotype. So an example, let's suppose you have UAC or UAU, and then due to either an error during DNA replication or maybe some mutagen, that UAU is turned into UAC. Well, if you look at UAU, that codes for tyrosine. If you look at UAC, that also codes for tyrosine. So even though there's a mutation, it hasn't affected the phenotype, and so we say it's a silent mutation. A missense mutation is where a different amino acid is inserted. The effect varies. So for example, let's suppose you have UAU, once again, that codes for tyrosine, but now it becomes UGU. Well, UGU codes for cysteine. So now we have a different amino acid. Now the effect of this can vary quite dramatically. In a lot of cases, a single amino acid that's wrong is not a big deal. You got to remember there's tens, hundreds, thousands, tens of thousands of amino acids in a protein. And so if you just have one little tiny change, for the most part, it's fine. However, there are cases where this is quite severe. For example, sickle cell anemia is the result of a single missense mutation in one key amino acid. And sickle cell anemia means that instead of nice kind of circular red blood cells, your red blood cells have this kind of sickle shaped. And the result of that is that they don't flow through the blood vessels as well. They can get clogged up. It can cause extreme pain. It's hugely problematic. We're going to talk more about that later on. But make no mistake, these can be a very big deal. It just depends. We also have nonsense mutations. These code for a stop codon. And so if you start again with UAU, but the mutation changes it to UAG, well, UAG is now a stop codon, which means that protein is just going to stop prematurely. And again, you could imagine that's probably going to be a pretty big deal if the protein is mid-build and all of a sudden just gets cut off. The final type of mutation is a frame shift mutation. A frame shift mutation is a change in what's called the reading frame of the translation. So remember, we read 3 codons at a time, right? So in a frame shift mutation, what happens is we either insert an amino or a base, or we delete a base. And so that's going to alter the 3-base reading frame and usually lead to a non-functional protein. So here's an analogy. Imagine that you have this sentence, Bob ate pie. So it's kind of like a codon, right? 3 amino acids, 3 amino acids, 3 amino acids. These are our codons. If we all of a sudden stick a little another letter in front, then our reading frame is thrown off. Instead of Bob ate pie, it's abo, bat, epi, and then we have this extra E at the end. And so same thing with reading the codons during translation. If you stick an extra base in front, it's going to throw off the reading frame, and every single amino acid downstream is going to be wrong. So that concludes our whirlwind tour of protein synthesis. Thanks.